All right, everybody. Good morning. We're going to get started. Just a quick reminder, or a couple quick reminders. You do have your first exam on Wednesday. So again, just a reminder, we are going to be kind of glossing over some things or skipping some things today. You are only ever responsible for what I talk about in class. If I don't talk about it, you're not responsible for it. So if you read some detail in your SmartBook assignments or something like that we did not talk about in class, you are not responsible for it. I try really hard to make sure the SmartBook assignments go over exactly what I want them to, but there are 25 chapters in this book, and it's impossible for me to catch everything, okay? But if you come across a detail or we skip over something that is in the lectures, but we don't talk about it, it is not going to be on the test. The second reminder is, and I say this with trying not to sound like I'm blowing you off. I'm not blowing you off, but if you have to miss class, remember that everybody is allowed, and I'm talking about regular lecture class periods. I'm not talking about exams, and I'm not talking about labs. I'm talking about regular lecture class periods. Everybody can miss up to 4 lecture periods without it affecting their grade at all. So you don't need to email me if you're going to be missing a regular class period. If you're going to be sick, or you just don't want to come, or you're whatever else, you don't have to email me. Okay? Remember, I have 150 students, so if everybody emailed me when they missed class, it would be a lot of emails. And that is why I do my attendance that way, is because everybody's going to get sick, everybody's going to have something come up, and that's fine. It doesn't affect your grade at all, okay? If there's some extreme circumstance where you're in the hospital for 3 weeks and you're missing extended periods of time, then yes, email me. But for just regular old missing class, you don't have to email me, okay? All right, so we are going to pick up where we left off, at least I think where we left off. Pretty sure. Okay, and we are going to start talking about the structures that you find in bacterial cells. Now, we're going to talk about all of these cells, or all of these structures, from the outside in. So what we're starting with, are the external structures. Okay, so the things that are kind of attached to the outside of a typical bacterial cell. Some of these things, we're going to kind of going back to this idea. Some of these things are things that you find in all bacteria, and some of these things are things that you find in most bacteria. Some of these things are things found in some bacteria. And I'm going to distinguish that as we go through, but keep in mind that that's something that you have to remember. Okay, so starting with the external structures, we're going to start with our appendages. And appendages, as a word, just means the structures on the outside of your body that help with some kind of function, right? You have appendages on the outside of your body. You have arms and legs. If you have a penis, that's an appendage too, right? They all serve different purposes, but they are external structures. They are appendages, and bacteria have appendages as well. Now, something important to keep in mind with most bacteria that we are going to talk about, and I know I've put it up here before, but bacteria and all other prokaryotes, for that matter, are unicellular. That means that the entire organism is made up of one single cell. And that means that it's one single cell floating around in whatever environment it happens to be in, whether that's inside a human body or in a body of water or on the dirt or on your desk or in somebody's ear canal. It doesn't matter. There is just one single cell floating around in the environment. So they have a lot of extra structures and appendages and things like that is designed to help them deal with that environment, okay? So things like flagella. Flagella are long, whip-like appendages. Okay, they're long and thin. This is an example of a flagella right here. This bacteria happens to have one flagella on either end. This bacteria happens to have, 4 or 5 kind of attached to the middle. And these are used mostly for motility, which is a fancy word for moving. So they whip these back and forth, and it helps them swim or move through their environments. The best example of this one that everybody knows about are human sperm cells also have flagella. A sperm tail is a flagella. And it whips around and helps the spermie get to where it needs to go. I'm sorry, I just called it a spermie. That was weird. A sperm where it needs to go. But bacteria have the same thing, right? They have flagella that whip back and forth and push them through the aqueous environments or water-based environments and get them where they need to go. Right? Next, we have pili, and pili are also long and thin, but they are tubular structures. And what I mean by that is they have like a hollow middle. They're like long straws. And their most important purpose is something called conjugation. And we'll talk about conjugation in more detail later, but conjugation is how bacteria exchange DNA. All right, now bacteria and other prokaryotes do not reproduce sexually. They don't exchange DNA in order to reproduce, but they do exchange DNA with each other. And we'll talk about why and what that looks like later. But just know for now that pili are used for conjugation, and conjugation is what is done to exchange DNA between bacteria. Okay, and this is an example of a pili right there. You can kind of see it. Right there. They're long little tubes. And two bacteria will attach to each other with their pili and exchange DNA through the tubes. Now, fimbriae are short and hair-like structures, and they are mostly used to help bacteria attach to each other. So all of these little short things sticking out from the edge of these bacteria, those are all fimbriae. They're short, hair-like projections, and they're mostly used to attach to surfaces or each other. Because again, remember that these bacteria are in whatever environment that they are in. And they have to be able to deal with that. So if they want to stay somewhere, they have to be able to grab on. A really good example of this, and not something that you have to write down, if I'm not writing it down, you don't have to, is a bacteria known as Neisseria gonorrhea. Okay, I can bet you can tell by its species name what disease it causes. It causes gonorrhea, which is a disease of the urogenital tract. Neisseria gonorrhea, the reason that they specifically, or one of the reasons that they specifically infect that area is because they have fimbriae that are especially good at attaching to the mucous membranes of genital tracts. They have special little hair-like projections that specifically can grab onto genital tissue. And so that is why they infect there, because that's where they're able to grab on and stay. And the rest of the body, not so much. Okay, so short hair-like projections. And the last one are called nanotubes, and they are very long, thin extensions. And you can kind of see them here. These are nanotubes, but they are very long and thin projections that are mostly used for energy exchange. So again, bacteria don't reproduce the same way that you think of animals reproducing, and they don't do sexual reproduction, but they do share materials with each other. Another way of kind of thinking of that is like forming a village, right? Each individual bacterial cell is one organism. It's like a little person. It's one organism. If you have a collection of bacteria, they can share resources back and forth. They can share energy and food to help everybody survive better, right? It's like living in a village where you share resources. You have a baker and you have a farmer and they swap, right? And that is how nanotubes work, or what their major purpose is sharing resources. All right, so after appendages, those are all the things that stick out from the outside of a bacterial cell. Next, we have the things that coat a bacterial cell. So we have the surface coatings. And we have two major types of surface coatings. We have an S layer and we have a glycocalyx. All of these things, by the way, all of these and all of these are sometimes structures, okay? Bacteria sometimes have these. Not always, not even most of the time. They sometimes have these. Some bacteria have them, some bacteria don't. Okay, so an S layer is a hard, thin layer of protein that kind of acts like armor. Okay? Remember again that bacteria are just expected to survive whatever environment they are in. Think about the way your human body works and the way that your cells are arranged. Okay? You are a multicellular organism. Most of the cells in your body are protected by the outer layers of your body. So your internal organs and all of your blood and everything that is held within your body, your brain, your spinal fluid, all that stuff is protected by your skeleton and by your outer skin layer. Bacteria don't have any of that. They're just floating around in the environment. Okay? The S layer acts like a layer of armor. It's hard and thin and protects them from things that might hurt them, just like why a human would wear armor. Protects you from being poked and hurt. It's the same thing. And a lot of times with S layers, bacteria produce them That's not a good color for that, is it? Okay. Bacteria will produce them when they are in a harmful environment. So if a bacteria is floating around in a nice water source and there's no potential, not predators, but no potential things that are going to hurt them, they have enough food, they have enough water, everything is great, they won't produce an S layer. Just like if I'm walking around and not in danger of getting in a sword fight at any moment, I'm not going to be wearing armor, right? If my body is not in danger, I'm not going to wear armor. The same thing is true for bacteria. But when they are introduced to a harmful environment, so like the inside of a human body is a pretty harmful environment for most bacteria. We have an immune system and all kinds of other things that are designed to destroy it. And so they will produce S layers that help them not be killed by the inside of the human body. Okay, and that's what makes it possible for some bacteria to cause infections. Not all of them, but some of them are capable of producing S layers and makes them able to get past the human immune system and get into our bodies and cause disease. And then our second surface coating is something called a glycocalyx. And a glycocalyx is another protective coating that bacteria can have. Remember, again, this is a sometimes thing. But this is a thick, slimy layer of protein and sugar. So rather than a thin, hard layer like armor, it's a thick, goopy, mucoidy, sticky layer. Okay? And that also protects it from things that might be floating in its environment. Similar to the way that we'll talk more about this later, but you also produce thick, slimy, mucusy stuff, right? It's called mucus. Your lungs produce it. And your lungs produce it. What happens when you're sick and you're coughing stuff up, right? That's your body getting rid of the stuff that it doesn't want. It's protecting the inside of your body from all of the crap that's trying to come in. That's what mucus does in your body, and it's the same thing here. The sticky, thick mucus protects it from things getting in and things hurting it. Okay? And the glycocalyx is also sometimes called a capsule. And you can see an example of a glycocalyx here. This is a picture of a bacteria growing on a plate, and you can see how shiny it is. It's what we call mucoid. It's very, very sticky. If you were to poke this with an inoculating loop, it would stick to the inoculating loop. Sometimes with these bacteria, you can touch an inoculating loop to it, and you can pull it straight up, and it'll string like a cheese pull. It's very, very sticky. It's mucoidy and thick. Okay. And you can kind of see the glycocalyx layer here. This middle part right here, that's the bacteria. The little lines in the middle, those are bacilli bacteria. And these areas surrounding them where there's no color, that's where the dye is not able to reach because of the glycocalyx. And we'll talk more about biofilms later, but biofilms are an important facet, I guess, when you're looking at bacteria and how they cause disease. Because, again, bacteria are single-celled organisms. But just because they're single-celled and one cell makes up an organism doesn't mean that they can't form communities, and they do. So biofilms are communities of bacteria that adhere to surfaces. And the reason that they're able to do that is through a glycocalyx. And you can kind of see in this picture here that the bacteria will form this glycocalyx, and it'll use that glycocalyx to kind of form this sticky, mucoid, thick, disgusting community. And this community will be very, very resistant to harm, okay? They like closed ranks, they form this giant glob of bacteria. It's basically like a community working together. They share resources, they work together to get rid of wastes, they work together to get food products. And so just like with humans, when people work together as a community, things go better for everybody, usually. They're more difficult to remove and or kill than any normal individual bacteria. And we all have firsthand examples of this. So when you wake up in the morning and your teeth feel disgusting, right? You run your tongue over your teeth and they feel bumpy and sticky, kind of thick and gross. It's because a biofilm of bacteria has formed on your teeth overnight. And that's one of the reasons that it's important to brush your teeth twice a day, is because if you don't, those biofilms will become more established. And as they become more established, they start causing gross breath, but they also will start to decay your teeth, they'll rot your mouth, they cause infections. And removing the biofilm from your teeth requires a certain amount of forcefulness, right? You have to physically scrape and brush the bacteria off your teeth because they've formed this community that can be harder to remove, right? Swishing some water isn't gonna do nothing. Even if you swish some mouthwash, that's not gonna do anything. You have to physically scrape it off. This is also true, algae can form biofilms. Have you ever seen the outside of a rock that gets really, really slippery because it's completely covered in thick, slimy, mucoidy algae? If you've ever tried, I know none of you are probably homeowners at this point in your life, but if you've ever tried to remove algae from cement, it's a giant pain in the ass because they don't want to come off, right? You have to pressure wash the absolute crap out of it to get algae off of cement. And that's because they're forming biofilms. Okay, and so this will come up more when we talk about infections. So the last whole portion of this class, it's always my favorite part. I like to think it's everybody else's favorite part too. But the whole last section of this class is talking about infections. We'll talk about the infections that bacteria cause, how they cause them. And there's a lot of bacteria that are particularly good at forming biofilms, and they are the most dangerous. All right, so next we have the boundary structures. And what the boundary is the line at which the inside of the cell becomes the outside of the cell. Okay. And again, there are going to be some things that we have here that all bacteria have, and some that only most bacteria have, and some that only some bacteria have. We're going to talk about it as we go through. Right? So in bacteria, we collectively call the boundary, we call it the cell envelope. And the cell envelope is made up of several different layers of different structures. And it depends on what type of bacteria we're talking about, what structures they have within their cell envelope. So again, the envelope is the collection of all of these structures. And this is kind of where gram-positive and gram-negative come back into play. So we talked about that last time, about gram-positive organisms and gram-negative organisms, and how about organisms are about half and half, right? Bacteria, you can tell a lot about a bacteria based on what they gram stain. And what gram staining is actually doing gram stains distinguish differences in the cell envelope. Okay? So we're going to look at cell envelopes as they are comparing gram-positive and gram-negative. So gram-positive organisms, as you can see over here, remember gram-positive organisms turn purple, and gram-negative organisms turn pink when they are gram-stained. The biggest difference, or I guess there's a couple of major differences, but the major substance in well, we're going to talk about this individually. I take it back. Okay, we're going to talk about the gram-positive and the gram-negative. So we're going to talk about the components of the cell wall. Now, the first component of the cell envelope, the first component of the cell envelope is the cytoplasmic membrane. Now, this can also be called the plasma membrane, or it can be called a cell membrane, right? It's given lots of different names. All of those names apply to the same thing. And the cytoplasmic membrane, remember, is one of those things that all cells have. Okay? So whether an organism is gram-positive or gram-negative, or if it's an animal or a plant or an archaea or a worm, it doesn't matter. They are all going to have some kind of cytoplasmic plasma cell membrane. Those words all mean the same thing. And just like we talked about before, cytoplasmic membranes are phospholipid bilayers. So you have this area where the hydrophilic head group and the hydrophobic tails arrange themselves so that the hydrophobic tails are tucked conveniently on the inside where they don't have to touch water because they're afraid of it. They don't like it. They're hydrophobic. So they get tucked neatly on the inside so that they don't have to touch the water. And what we end up with is this layer of phospholipids with the hydrophilic part on the outside, hydrophobic part in the middle. And this phospholipid bilayer is what we call fluid. And what that basically means is this is not a rigid structure. A phospholipid bilayer is flexible, okay? Maybe that's a better word. Both of them apply. But they're fluid or flexible. They can wave. They are very adaptable to shape. Okay? They're soft. They're not rigid structures. Another important characteristic of cytoplasmic plasma cell membranes is they're what we call semi-permeable. Now, that's just a fancy name. If you look at the word permeable, permeable means things can get inside. If something is permeable, it means stuff can get in there. Semi means kind of. So what this means is that some things can enter a cell, some can't. So this phospholipid bilayer is what makes up the boundary of every single cell. It is what marks the inside of the cell from the outside of the cell. And the semi-permeable membrane is responsible for keeping some stuff out and letting some stuff in. Stuff that you don't want to be in your cells, you keep out. Some stuff you can let in. Some things that can enter are going to be small molecules like water and electrolytes. Some things that can't are going to be basically everything else. Proteins, sugars, fats. Okay, so waters and electrolytes are very, very tiny molecules. Electrolytes are just one element big, and water is just a couple of elements. They're very, very tiny. A lot of those can cross freely across the membrane. They can go in and out at will. Sometimes, most of the time. Other molecules like carbohydrates, proteins, DNA, lipids, those are all macromolecules, right? They're big. That's written in their name. And they cannot cross membranes freely. We're going to talk more later about how they get into cells, because they do need to. But most of the time, they need help. What this means is that the bilayer can basically pick and choose what it wants to let across. So if a cell needs sugar, it will let sugar across. If it needs certain proteins, it will let proteins across. But it gets to make those choices. Right? And remember that all cells have that. Now our next layer of the cell envelope is a cell wall. And this is a structure that most bacteria have. Okay. Most bacteria have a cell wall. There are very few examples of organisms that don't. Most eukaryotes don't have one. We'll talk more about that when we get to chapter 4. But most bacteria have a cell wall. And most bacteria that have a cell wall made out of this molecule right here called peptidoglycan. And you might be able to tell from the word peptido, like a peptide, is a protein. Glycan is like glucose, and it's a sugar. So peptidoglycan is a protein sugar molecule that most bacterial cell walls are made of. And kind of an interesting note, I think this is interesting, but is that the only place that you see peptidoglycan as a molecule is in bacterial cell walls. So you don't see it anywhere else in nature. You don't see it in plants or algae or funguses or anything else. The only place you see peptidoglycan in nature is in bacterial cell walls. And remember my language here, most bacteria have cell walls, and most bacteria that have cell walls have peptidoglycan in their cell wall. And so this is the one that we're going to remember because we, in this class, are dealing with the most common scenario. But in this case, there are bacteria that don't have cell walls. Some of them are very, very difficult to treat because they don't have cell walls. And some of them have cell walls that are not made out of peptidoglycan. A really good example of that that's really hard to treat is tuberculosis. Tuberculosis is a weird bacteria that has a cell wall not made out of peptidoglycan. What it's made out of, we're not going to worry about right now. But again, this is most bacteria have cell walls. Most are made out of peptidoglycan. Okay? And of the bacteria that have peptidoglycan in their cell walls, so of the bacteria that have cell walls and those that have peptidoglycan in their cell walls, they mostly fall into one of two categories, which we've talked about a little bit. They're either gram-negative or gram-positive. And they are differentiated based on the structure of their cell wall. Now, gram-positive bacteria, their peptidoglycan layer is very, very large. Okay, so you can see in this diagram here, and if you go back to the other diagram I had a couple pages ago, their peptidoglycan layer is very, very thick. They have a thick peptidoglycan layer. As compared to gram-negative bacteria, they have a very thin layer of peptidoglycan. So gram-positive have a thick peptidoglycan layer. I'm going to abbreviate that. PDG is peptidoglycan. And gram-negative have a thin PDG layer. They both have the same molecule. They both have peptidoglycan. One has a thick layer, one has a thin layer. And then the other biggest difference is that notice that both of these cells have a plasma membrane, right? Since all cells have a plasma membrane. So they have a plasma membrane on the inside, closest to the inside of the cell. So this is the cytoplasm, the inside of the cell. You have the cell membrane, and then you have your peptidoglycan layer. So the cell wall is actually on the outside of the plasma membrane. The gram-negative bacteria have an extra phospholipid bilayer. It's called the outer membrane. So while both organisms have a plasma membrane, right, they both have one layer of phospholipid bilayer. Gram-negative rods have an extra one. There's none up here for gram-positives. Okay. So gram-negative ones have an outer membrane. Okay. And those two structures, and we're not going to talk about in this class, again, why the one with thick peptidoglycan stains purple and why the one with thin peptidoglycan stains pink. You will absolutely have to know that if you're in lab. We'll talk about it more later. But in this class, you need to know these differences that I have written here, that gram-negative bacteria have a thin peptidoglycan layer and an outer membrane, and that gram-positive organisms have a thick peptidoglycan layer. And you have to remember that gram-positive bacteria stain purple, and gram-negative bacteria stain red or pink, which we talked about before. I always think the way that I remember this one is that if you think about the layer of peptidoglycan like a sponge, the thicker layer of peptidoglycan is going to hold color much better, which means that the gram-positive ones will be dark, right? So that's how I remember it in my head, is that the gram-positive ones are dark because their thick peptidoglycan layer soaks up all that color so much better. It's more complicated than that, but that's kind of the gist of it. And that the thin layers of peptidoglycan don't hold on to color as well, so they turn a lighter color of pink or red. Okay. All right, so that outer membrane, we're just going to talk about that briefly, is an extra membrane on the outside of a peptidoglycan layer for only gram-negative bacteria. Remember that gram-positives don't have it. Gram-negative bacterial cell walls have a cell membrane, a layer of peptidoglycan, and then an extra layer on the outer membrane. A lot of times, this provides extra protection for the gram-negative bacteria. Gram-negative bacteria are much more difficult to kill with antibiotics than gram-positives because they have that extra layer of membrane. And that outer membrane is also capable of producing toxins, which we'll talk more about later. But a lot of those toxins are what cause symptoms inside of our human bodies. So a lot of gram-negative bacteria will produce these toxins that make you vomit or make you have diarrhea or make your kidneys not work anymore, things like that. What's your question?
Do all gram-negative cells have the outer membrane?
Yes, all gram-negative cells do have an outer membrane. Mm-hmm. Now again, remember that is all bacteria that have a cell wall and have peptidoglycan in their cell wall. If a bacteria doesn't have a cell wall or doesn't have peptidoglycan in their cell wall, this doesn't apply. So, but all bacteria that meet those conditions, the gram-negatives all have an outer membrane. The structure may be slightly different, but they will all have one. All right, so now we're going to talk about our internal structures. We talked about the stuff that kind of sticks out the sides. We talked about the boundary, the cell envelope. Now we're going to talk about everything that's inside. And this is kind of where I'm going to probably skim a lot of stuff because we got to get rocking on this. I spent too much time talking earlier in the class. So just keep that in mind as we go through. So the first internal structure is the cytoplasm. Now the cytoplasm, again, is something that all cells have. This is the gelatinous solution encased by the cytoplasmic membrane. So it's just the stuff inside the cell that fills out the middle of the cell. It's like a watery kind of gelatiny substance. And it's typically a mix of water and other various nutrients, sugars, lipids, proteins, salts. And then any other internal structures like ribosomes, genetic materials, all the stuff we're going to talk about in a second will float around in the cytoplasm, at least in bacterial cells. Okay, so everything else that we're talking about will just be sitting in the cytoplasm. Okay, and there's just another kind of picture of all of this, that the green stuff in here is the cytoplasm, all that green stuff in the background, and then everything else is just kind of floating in there. So for bacterial genetic material, now remember that genetic material, again, is something that all cells have. But bacterial genetic material looks a little different than eukaryotic genetic material. And we're going to spend a fair amount of time talking about the differences between those. Now, the biggest difference is the one that sets prokaryotes and eukaryotes apart from each other. And that's that bacteria do not have a true nucleus. Because bacteria are prokaryotes and they do not have a true nucleus. Never. Okay. Eukaryotes have a true nucleus, and we'll talk about that more when we get to chapter 4. But bacteria usually have an area within their cytoplasm called the nucleoid, and that is just the area where their genetic material is held. And bacterial chromosomes, the word chromosome is just another name for their genetic material. Now in bacteria, for the most part, there is usually one circular chromosome. Okay, we talked about before that DNA, we talked about this briefly, that DNA is generally a double helix, and that's true in bacteria as well as eukaryotes. That's true for everybody. But in bacteria, they tend to have one double helix, one single one, and it tends to be in a circle. So that double helix loops back on itself and it attaches and forms a ring. Now that's in contrast to eukaryotes, who usually have multiple chromosomes, which I have written down later, but they usually have multiple chromosomes and they're usually in a straight line. So for example, in humans, we have 23 pairs of chromosomes. So we actually have 46. Bacteria have one, and it's a circle. But something that bacteria have that eukaryotes don't, as far as I know, eukaryotes don't carry these at all. There's probably some exceptions, but as far as I know, this is not a thing for any kind of eukaryote. This is something special that bacteria have that the rest of us don't. And they are something called plasmids. And what plasmids are is they are little extra bits of DNA. They're not essential. They're not required for reproduction or anything like that. They're not required. They're just extra pieces. But what these do is they carry extra genes that provide advantages. Okay, and those advantages can be anything from making them adhere better to certain surfaces or producing certain toxins that make them better able to infect a human or anything else. They provide extra little copies of traits. And this sounds ridiculous when I say it like this, but the kind of thing here that bacteria are actually able to do, they're actually able to do this, but when you put it in human terms, it sounds absolutely ridiculous, is it would be like me saying, I have blue eyes. It would be like me saying, I would like to have brown eyes. So can somebody in this classroom who has brown eyes hand over a piece of their DNA so that I can have brown eyes too? Right? If that's what I wanted to have, I wanted that advantage. I wanted the ability to run faster. So if somebody in here who runs faster than me could provide some DNA that makes me run faster, that would be great. Right? But that is what bacteria do. They have these little extra genes that make them resistant to antibiotics or produce a certain toxin, and they can exchange them through pili. They can say, oh, this gene is really going to help you survive better, have a copy. And then that other bacteria can pick it up and have that gene. Okay, so that is absolutely, of course, not something that humans are capable of. If that ever becomes a possibility, it will be called gene therapy, and it will be a medical marvel. But bacteria do it all the time. And it's part of the way that bacteria are able to mutate so quickly and change their genes so quickly. We'll talk more about that as we get to reproduction later. But that is what plasmids are. They're just little extra bits of DNA. And a really good example of a bacteria that does this, and you don't have to write it down now, I think I have it later, but there are two bacteria, there are two species of bacillus. One species is called Bacillus cereus. One species is called Bacillus anthracis. Now, Bacillus cereus causes a very mild case of food poisoning. Okay, it's going to make you hug the toilet for 12 hours, and then you're going to be fine. Most people don't die from it. Okay, it's a miserable 12 hours, but then you're going to be fine. Bacillus anthracis causes a debilitating illness that shuts down your entire immune system and kills about 75% of the people infected with it. Okay, that's a big difference, right? The only difference between those two bacteria is two plasmids. 99.9% of their DNA is exactly the same, but those two extra plasmids make Bacillus anthracis one of the most deadly infections in the world, and Bacillus cereus one of the most mild. Okay, so plasmids provide incredible advantage to bacteria. Right, next we have ribosomes. This is another internal structure that's just kind of floating around in the cytoplasm of bacteria. And ribosomes are the site at which proteins are made. So synthesis means made. So ribosomes are the structures that make proteins. And again, ribosomes are one of those things that we said all cells have. All cells have ribosomes. And again, all living things, all cells have some type of ribosome. But what the ribosome looks like is going to be slightly different in between different species and domains of organisms. In bacteria, they have a relatively small ribosome. And we're not going to talk much about what this term means, but this is one of the few numbers that you have to remember. So bacterial ribosomes are small, and they are what we call 70S ribosomes. In eukaryotes, which I'll have on a slide in a little while, they are 80S ribosomes. Okay, 70 is smaller than 80. That's a unit of measurement. So they are 70S. They're small relative to eukaryotic ones. And they are also free in the cytoplasm. When we talk about eukaryotic cells, we'll talk about how ribosomes are embedded in what's called the endoplasmic reticulum. Okay, they have a particular spot that they're held inside of a eukaryotic cell, but in bacteria, they're just free floating. Okay. That's all you really got to know about ribosomes for now. Okay, we're going to skip this slide. I just don't want to talk about it. Inclusions we're skipping. All right, next we have endospores. And again, this is something that some bacteria are capable of producing. This is not something that even most bacteria are able to do. It's a very specialized process that only a select few, some amount of bacteria are capable of producing. And what endospores are, is they are a hardy structure that helps bacteria survive extremely harsh environments. Okay? It's a protective structure. And basically what happens is if a bacteria is put in an extremely harsh environment, like somewhere where it's really hot, or somewhere where there's no water, or somewhere where there's tons and tons and tons of chemicals, things like that, they will basically form a hard shell, kind of like a seed. It's not exactly a seed, but it'll form a hard shell around its DNA. And when this endospore forms, and you can see this endospore right here, and this endospore, when the rest of the cell dies, that DNA is going to be protected in this little seed. And kind of similar to other seeds, this endospore can sit around in the environment until it is no longer dangerous. And it will sit there dormant. It's kind of what we call dormant, so it goes to sleep. It's not technically living at that time, just like a seed, right? Like a plant seed. It's dormant, and it can sit there in the environment until the conditions stop. And when the conditions become favorable again, the endospore will germinate and the bacteria will come back alive. Okay, so you can imagine that this is an incredibly, incredibly advantageous process. Endospores are extremely resistant to temperatures or to any kind of harsh environment. You can boil endospores for days at a time and they will not die. You can expose endospores to straight Lysol and bleach and they will not die. You can expose them to radiation at some level and they're not going to die. All of the ways that we use to kill bacteria basically don't work for endospores. There are a few ways that you are capable of killing endospores, which we'll talk more about when we get to control of bacteria. But endospores allow a bacteria to survive incredibly harsh conditions: boiling, freezing, radiation, bleach, drying, all of those things. Okay. And the two genera, that's the plural of genus. That word weirds me out. I don't know why. But genus is singular, genera is plural. It doesn't sound like it should be the plural, so I feel like people get confused when I say it. But the two genera of organisms that are capable of producing endospores, for the most part, are Bacillus and Clostridium. Now, one that you may have heard of before is a Clostridium called Clostridium botulinum. Clostridium botulinum causes botulism. And the reason that it causes botulism, it mostly causes it in canned foods. Food canning is an extremely harsh process. A lot of time you're adding tons of salt or tons of sugar, you're boiling it, you're heating it, you're sealing out all of the air. Most things are not going to survive in that. However, Clostridium bacteria, like Clostridium botulinum, can. And it does. And that is how some cases of botulism food poisoning happen. Clostridium botulinum is also capable of living inside of honey, which no other organism is really capable of doing. If you've ever noticed, honey basically doesn't go bad. It doesn't grow mold, it doesn't grow bacteria or anything like that, but it can grow botulism, which is why you're not supposed to give honey to anybody under the age of 1, because their immune system can't handle it. But if you didn't know that, when you have babies someday, if you haven't ever had babies, don't give them honey. You're not supposed to. Okay, those are endospores. Now we're just briefly going to talk about archaea. Like I said before, archaea are structurally kind of similar to bacteria, but they are actually more closely related to eukarya. So we're actually going to look at just the slight differences between bacteria and archaea, but do remember that both bacteria and archaea are prokaryotes. So do archaea have a nucleus? Anybody? I see all you shaking your head. No, they don't, because they're prokaryotes, so they do not have a true nucleus. Okay. So archaea, like I said before, are structurally similar to bacteria. They do have they do not have a true nucleus, so no nucleus. Most of them do have a cell wall, but that cell wall is not made out of PDG. Ooh, that's not how you spell that. We're not going to talk about what it's made out of. You just have to remember that it's not peptidoglycan. So bacteria have cell walls made out of peptidoglycan. Archaea have a similar structure. It's a cell wall, but it does not have peptidoglycan, as we have written here. No peptidoglycan cell wall. And they are also capable of producing capsules, the glycocalyx layer, flagella. They have ribosomes, just like all other cells. They have a plasma membrane, right? And this cell, this archaea cell, looks very similar to a bacteria. They're structurally very similar to bacteria. Their biggest, the thing they're most known for is being extremophiles. And what I mean by that is they tend to grow in extreme environments. They are the organisms that can grow in Arctic ice and almost frozen water or frozen water. They can live deep in the ocean where it's extremely cold or extremely hot, depending if you're next to a lava vent, where there's no light and no oxygen. They can live in environments like a hot spring, right? And this is a hot spring somewhere. That yellow-green color in the inside of this hot spring is produced by the archaea that's growing in there. Okay, most bacteria can't survive near boiling temperatures, but archaea can. They can grow inside of lava. They can grow where lava has just run off. They can grow in the ocean, inside of Arctic ice, places where not many organisms can live. And as of right now, there are not any archaea that are known to cause human disease. Okay. There may be some that exist that we don't know about them, but as of right now, there are no known archaea that cause human disease, which is why we don't talk about them much. They're extremely important to the ecosystem. If you're ever curious about archaea, you can go talk to Dr. Wells in the biology department. He works with archaea, and he's doing some crazy stuff where he's making them breathe gold and producing cancer-curing nanoparticles. It's wild. So they are extremely valuable. They control a lot of our environments, but they don't cause human disease, so we kind of skip over them in this class. Yeah. I'm so okay, sorry. All right, so there's a lot of information on this slide. I'm gonna point out the things that you have to remember, okay? And this is just a comparison between bacteria, archaea, and eukaryotes, which we're gonna talk about in just a minute. The first one that I wanna note is that both bacteria and archaea, in terms of their chromosomes, they have single circular chromosomes. So bacteria and archaea are similar in that way. They don't have a nucleus, and their chromosomes are generally circular and single. So one circular. Archaea usually have multiple linear chromosomes. All right, the next thing I want you to notice is that bacteria, like we talked about, have 70S ribosomes, which means their ribosomes are smaller. 70S. Eukarya have 80S. I know we didn't talk about that. We did kind of. Eukarya have 80S ribosomes, which means they're slightly bigger and slightly more complex. Now, archaea are kind of tricky. They have 70S size ribosomes. So they're 70S size, so they're smaller, but they function like 80S. Okay, so they're kind of in between the bacteria and the archaea. Their ribosomes are smaller, like bacteria, but their ribosomes function very similarly to eukaryotes. And we're not going to get into really what that means, the function of ribosomes. It's very complicated and technical. All you have to know is that the archaea ribosomes function more like a eukaryotic one, even though they're smaller. All right. The next one is that the only organisms that have cell walls made out of peptidoglycan are bacteria. Archaea, most archaea have cell walls, but not made out of peptidoglycan. Okay. And eukaryotes can have cell walls, but they're never made out of peptidoglycan, and most of them don't. And then the last one, which we've kind of talked about, is that eukaryotes are the only ones that have a nucleus and membrane-bound organelles. We're going to talk about what membrane-bound organelles are in a minute, but we've already talked about what a nucleus is. And eukaryotes are the only ones that have a true nucleus. Right? So that is the basic structure of a bacterial prokaryotic cell. Okay? So now we're going to move on to chapter 4. We're just going to keep blazing right through. And chapter 4 is looking at eukaryotic cells and microorganisms. Now, remember again that eukaryotes is a very broad term that applies to many different types of organisms. Eukaryotes are plants, animals, funguses, algae. There are certain protozoa. All of those organisms, right? That's a huge diversity of organisms are eukaryotes. In this class, we're only going to be talking about 3 major groups, okay? Because plants, for the most part, are not microorganisms. Animals, for the most part, aren't, but there are a couple that are. So these are the 3 major groups that we are talking about. But I don't want you to think that these are the only 3 groups of eukaryotes, because they're not. Eukaryotes, again, are plants, animals, funguses, protozoa, protists, all different kinds of stuff. These are the 3 we're going to be talking about. And it's because these are the three that are mostly responsible for affecting human health. Okay. While plants are incredibly important to human ecosystems, they don't typically affect human health all that much. There are some that are poisons, but they don't infect us. Okay. So the first group are the fungi, or the funguses, fungi. And this includes the yeasts and the molds. And we're going to talk about the differences between those later. Protozoa is our second group, and these are single-celled, kind of what we typically call primitive animals. They act like animals, but they're single-celled. So they move like animals. A lot of parasites are these protozoa. They're single-celled organisms. Amoebas, things like that are protozoans. So we'll talk about those. Again, we'll talk about more of these in detail. And then the last group are helminths, and helminths are just parasitic worms. Okay. Now, helminths are actually, they're worms. They're just like regular worms. They are animals. Okay. So they're just like our regular worms that we think about, like an earthworm. It's just that this group, this helminth group, typically invade other organisms. Like earthworms don't cause human disease because they don't live inside of humans. But these helminths do. They can and they do. They live inside other organisms. We're going to talk about the general structure found in eukaryotes, and then we'll move on to specific characteristics of these three groups. All right, so just like last time, we're going to be talking about the general structure of some of these eukaryotes. We'll differentiate structures that all eukaryotes have from structures that some eukaryotes have. And you can see it up at the top. These are things that are found, I think this, whoops. Is this thing that all eukaryotes have? Okay. I couldn't remember how this picture worked. So almost everything on this picture is something that all eukaryotes have. So all this stuff across the top, ribosomes, membrane, microtubules, mitochondria, nucleus, different endoplasmic reticulums, up until all this up part is things that all organisms, all eukaryotes have. Okay. And then these things at the bottom are some things that some eukaryotes have. If you ever get confused, you can come back to this slide. All right, so just like last time, we're going to start with the external stuff and then move our way in. Okay, so just like last time, we talked about appendages. Appendages exist in eukaryotic cells too. I already gave you one example, sperm cells. Sperm cells are eukaryotic cells. They have flagella. But there are other types of organisms that have flagella. But flagella, again, are long and whip-like. And they're typically used for cell motility. So flagella are more common in cells that need to be able to move. Think about the cells in your human body. The cells in your body don't usually need to move for the most part. Things like sperm do, but most of the other ones, like my kidney cells, don't need to swim to my heart, right? They need to stay where they're supposed to be. But single-celled eukaryotes, like certain parasites and things like that, do need to move, and they have flagella. So we talked about this little guy last time. This is Trichomonas. Trichomonas is that vaginal parasite that I told you about last time that survives in human vaginal tracts and swims around in urine. It swims around because it has these little flagella. You can see it has like between 4 and 5 flagella on each cell, and it will swim through vaginal tracts. And most people get infected with Trichomonas either through direct sexual contact, but you can also get Trichomonas from like hot tubs because they swim through the water, which is real gross, but it's true. All right, and then these are sperm cells. You can see the sperm head, and then there's this little flagella tail sticking out the back. Okay, so they're mostly used for motility. Now, cilia are kind of similar, not exactly, but they're similar in structure to fimbriae. These are short, hair-like cells. And they can be used for either cell or environment motility. Now, what I mean by environment motility is these short little hair-like, and these are cilia over here. This is an example, these little short hairs sticking out of the top of those cells. This is a picture of, I think this is a lung tissue. And on the outside of that lung tissue, there's all those little cells, those little cilia sticking out. And this is the outside of a eukaryotic parasite. You see all those little hairs sticking out. So you can use cilia to move, like this organism, this is called a paramecium. You don't have to know that name. But it's an organism that swims through its environment. It's a single cell, and it uses all of those little hairs to wave itself around. It's a very gentle, like rocking motion. But inside organisms like humans, we also have cilia. But the cilia that we use mostly move the environment around the cell. So a really good example is inside your respiratory tract, you have cilia that moves the mucus along. Okay, your cells don't move, but they have little cilia that wave the mucus along. Inside your gastrointestinal tract, you have cilia that move the food along. Inside your fallopian tubes, if you have those, you have little cilia that move the egg where it needs to go. Okay, so cilia can be used for cell motility, but they're also used for environment motility, for moving other things where they need to go. And I forgot to tell you this on the other slide, but down at the bottom, the other slide that had appendages as well, bacterial ones, there's a couple of links to YouTube videos where you can see them moving, if you want to see them moving. You can copy those links. Okay, I kind of think of the movement of cilia moving its environment of like somebody crowd surfing at a concert, right? Somebody sits on top and everybody moves their arms in one direction and somebody will move across the crowd. All right, so next we have our boundary structures, just like last time. These are the things that separate the inside of a eukaryotic cell from the outside. Just like everything else, all cells have a cell membrane, which is a typical phospholipid bilayer. It is semi-permeable, just like we saw before. Now, the big difference between bacterial cell membranes and eukaryotic cell membranes is that eukaryotic cell membranes tend to have things called sterols. Now, sterols are lipids, and these sterols provide structure. Now, the reason that most eukaryotes need these and bacteria don't is because most eukaryotes don't have a cell wall. So think about a bacteria cell membrane. Remember, we talked about it as a very fluid, flexible, liquidy thing. A phospholipid bilayer by itself has very little structure. It's very fluid. It can fall apart. But in most bacterial cells, they have a layer of peptidoglycan around the outside that acts to protect it. And the cell wall is what provides structure. The cell wall is like a hard structure on the outside of the membrane that holds it together. We have this whole extra layer of protein and sugars that glue everything together. Eukaryotic cells don't typically have that, okay? Some do, I don't know, I don't want to write that. But it's more common for eukaryotes not to have any cell wall at all. What's up?
Is there a difference between selectively permeable and semi-permeable?
No. Well, sort of. But we'll get to that when we get to transfer stuff. Yeah. He asked if there was a difference between selectively permeable and semi-permeable, and the answer is kind of, but not really. I think for now, just think of them as the same thing. When we get to transfer, we'll talk about the slight difference. But cell membranes, I'm getting my words confused, cell membranes on their own do not have a solid structure. So we introduced things called sterols to help hold everything together. So I think of sterols as like glue, right? Sterols are kind of stuck in the lipid membranes to hold them together. So sterols, in humans, the most common sterol, our most common sterol is cholesterol. So cholesterol is something that gets a really bad rap, right? We all hear about it in terms of heart disease. I guess most of you are probably not at the age where your doctor is yelling to you about your cholesterol, but most of us will get there. But cholesterol gets a really bad rap, right? You don't want to eat too much cholesterol. It's going to give you a heart attack by the time you're in your 40s, all that stuff. You don't want to eat bacon because it's got too much cholesterol. Cholesterol is absolutely necessary. You need it. You need it to hold your cells together. If you didn't have any cholesterol, your cells would literally dissolve. They would not stay together. They are necessary to provide the glue. The problem is if you have too much cholesterol, your cell membranes become too stiff because your body doesn't have anything else to do with those cholesterols, so it starts sticking it in all the cells that it can find, and eventually those cells will get too hard, right? So there's a happy medium here. We don't want a cell membrane that's so loose that it's going to fall apart and so fluid that it has no structure, but we don't want a cell membrane that's so rigid that it doesn't move the way that it's supposed to. Okay, so that is what sterols do. They provide structure. All right, now there are some eukaryotic cells that have a cell wall. Some. Particularly plants and algae and fungi. Okay, so within eukaryotes, the only real organisms that have cell walls around their cells are certain types of plants, or most plants, certain types of algae, and certain fungi, or all fungi. The only organisms that we are going to focus on are funguses, right? Because again, plants and algae do not typically cause human disease. So while those cells do exist, we're not going to talk about them much. So we're only talking about fungi. And fungi, their cell walls are made out of something called chitin. You do have to remember that name. Funguses, yeasts, and molds have cell walls made out of chitin. And chitin is the substance, it's a very hard, rigid substance that's found in the exoskeletons of beetles. That's the same thing. So that thing, that crunchy sound you hear if you step on a beetle, that's caused by the chitin in their exoskeleton. And that is what the cell walls of funguses are made out of. It's a very hard, rigid structure and provides a lot of protection and strength for these organisms. Right, next we're going to talk about the internal structures. And the first one, the kind of big one, is the nucleus. And the nucleus, this is a picture of a nucleus. This is kind of zoomed in on where the nucleus is within a cell. The nucleus is a structure whose main function is to store and separate the DNA from the cytoplasm. So the nucleus of a eukaryotic cell is separated by a whole extra nuclear membrane. Okay, it has its own phospholipid bilayer. And what it actually has on a nucleus is 2 layers of a phospholipid bilayer. So a nuclear envelope has 1 layer of phospholipid bilayers and 2 layers. There's actually 4 extra layers of phospholipids. Okay, and the DNA is all packaged inside of the nucleus. It's kept separate from the rest of the cell, and this affects how a cell reproduces and uses its DNA. We're going to talk more about that later, I think in the 3rd section. 2nd section? I don't remember. We're going to talk about it later, what the effect of that is, why that matters. But for now, we just need to know that this is true. And these bilayers, this extra nuclear envelope, this whole separate membrane that separates the DNA from the rest of the cell, will have these things called pores on them, which are basically just little holes, which means that it lets very specific things in and keeps everything else out. All right, and then the nucleolus, which is named down here is a very specific area within the nucleus where the DNA is. Okay, so in this picture here, all of this out here is cytoplasm. This yellow line right here, or yellow, this red line right here is the nuclear envelope, and then this blue part is where the DNA is kept, inside the nucleolus. Now, the genetic material of eukaryotes, we kind of touched on this a little bit, but I want to revisit. The DNA of eukaryotes is packaged into these very, very, very condensed chromosomes. Condensed chromosomes made up of DNA wrapped around histones. So we take these DNA double helices and basically wrap them in circles around a bunch of proteins. The way that they've always described this to me, I think is weird, but it's basically like taking a string of pearls, I guess, and then wrapping the DNA around all of these little balls, okay? And then all of those little balls are stacked on top of each other in a really nice little organized way so that all the DNA inside of every single eukaryotic cell can be packaged down into a teeny tiny little package. One of the things, and again, you don't have to know these numbers, but it's just to kind of put it in perspective. The DNA inside of a eukaryote is so tightly condensed and coiled that if you took the DNA from a single human cell, so one cell inside of a human, and you uncoiled it and stretched it out, it would be 6 feet long inside of one of your cells. And think about one of your cells is about 1 millimeter big, even a little bit smaller than that. But if you stretched out all that DNA, it would be about 6 feet long. Instead, it fits inside of a nucleus, which is about 5 micrometers big. Okay, so the difference there is huge. I think that there's some saying somewhere that, like, if you took the DNA inside of a single human and stretched it out from every cell, it would reach to the sun and back. Like, that's how big the DNA is, but it's condensed in this teeny tiny little package. And what that allows is it allows us to have more DNA than bacteria. More DNA means more genes and typically means more complexity and able to accomplish more things. Now, again, in general, eukaryotes are going to have multiple chromosomes, not just one, like you see in bacteria. And those chromosomes are going to be linear. They're going to be in straight lines. Now, not every single one, but eukaryotic organisms can have various numbers of chromosomes. Most animals don't have the same numbers as each other. For example, humans have 23 pairs. And this is a picture, this is called a karyotype. This is a literal picture of chromosomes inside of a human. Okay, so we have 23 pairs, we have 46 altogether, but you can see that they're straight lines, they're not circles, and there are multiple of them. The exact number of chromosomes will vary from species to species, though. So it's not like every organism has 23, but every eukaryotic organism will have multiple chromosomes. All right, and I think that is where we will call it today.